Now that I have confessed myself to be a skeptic about quantum entanglement and non-locality playing a role in brain function, I must also express skepticism toward my own skepticism. I'll now give concrete reasons that might end up proving my skepticism to be just plain wrong. As a first example, it has been shown that even massive molecules such as carbon-60 or fullerene exhibit interference effects when passed through a double slit, one molecule at a time. By comparison, neurotransmitters are typically much, much smaller than this, and the ions that pass through ionotropic receptors are even tinier still. The diameter of an open NMDA receptor pore is about 5.5 angstroms. By way of comparison, slits that are orders of magnitude larger generate interference effects when electrons are passed through them. Given their small size, NMDA receptors can be packed together much more closely than this, which, if simultaneously open, as occurs during bursting, would create the conditions for two or multi-slit interference. So certainly, wave-particle effects could, in principle, be expressed among neurotransmitters in the synapse. The key question is whether such effects matter in the functioning of a neuron. I'm skeptical, but I admit I don't know. If so, this would be another mechanism for introducing indeterminacy from the quantum domain to the level of variability in spike and burst timing. Another known mechanism can be found in the retina, where a cascading avalanche of signals can follow the detection of even a single photon. How can sensors in the retina detect a single photon? If there was ever a quantum domain event, it would be the absorption of a single photon. Evolution has come up with a protein molecule called rhodopsin, which is in the photodetectors in our retina. The key subcomponent of this is called retinol, which is basically very close to vitamin A. So when people say vitamin A helps you see, there's a good bit of truth in that. Retinol is amazing. It changes its shape if a single photon is absorbed by it. It changes its shape from this to this. Thus, the central foundation of our most important sensory system, vision, and ultimately of our visual conscious experience, is the isomerization and consequent shape change of a molecule derived from vitamin A. A single photon of light isomerizes a single molecule of retinol bound to rod or cone opsins. This is then amplified through a cascade of downstream processes that greatly amplify the signal, which can ultimately increase the likelihood that a neuron in the retina, called a ganglion cell, will fire and tell the rest of the brain that light has been detected at a certain location on the retina. This is amazing, and it is a quantum domain process that plays a central role in the most important aspect of our perception, namely seeing. Other scientists have recently argued that the perception of smell is similarly rooted in quantum domain processing. Now, things get even stranger in the retinas of birds. Some scientists have recently argued that migratory birds have a kind of compass in their retinas that allows them to detect the orientation of the Earth's magnetic field lines. In particular, there's a cryptochrome protein called Cry4 expressed in the cones of migrating birds, which many think is likely to be the magnetoreceptive protein used by birds. But it is not magnetoreceptive because it contains anything like tiny bits of iron that are magnetic. Instead, it detects light. According to the so-called radical pair model of the avian magnetoreceptor, it depends on long-lived electron spin coherence or entanglement. Now, you've probably heard the term free radical. Physicists have long known that the rates and yields of reactions involving free radicals are influenced by the application of magnetic fields, a phenomenon that they call the radical pair mechanism. Evidence is accumulating that migratory birds sense the direction of the Earth's magnetic field by means of radical pairs formed by the influence of detected photons on the cryptochrome protein Cry4. Sensitivity to magnetic field orientation arises because of the radical pair's spin dynamics. In particular, it depends on the spins being coherent across the pair. So evolution has come up with a protein that effectively shields coherent spins from decohering, despite the warm noisiness of the bird's retina, and this coherence or quantum domain entanglement allows migrating birds to see the orientation of magnetic field lines. It's incredible. Now, whether birds actually consciously experience the orientation of the magnetic field lines of the Earth within associated qualia, say north looks reddish and south looks greenish, I have no idea. I would not even know how to find out the answer to the question of what it's like to see magnetic field lines. But even if this mechanism has nothing to do with consciousness, if this model of avian magnetoreception in the retina is not ultimately falsified, it will be, in my opinion, the first strong evidence of quantum domain entanglement playing a central role in brain processing. Beyond brains, it may be that quantum domain entanglement plays a central role in other biological processes. Numerous experiments in recent years have supported the conclusion that photosynthesis is afforded by quantum domain effects. Scientists noticed that coherence within the light-capturing antenna proteins of green sulfur bacteria lasted much longer than one would expect, given the decohering influences of the warm temperatures they exist in. Here's a picture of the protein complex of these bacteria. The gray areas are protein scaffolding, and the green areas are the chlorophyll proteins. Once a photon is captured, it triggers electron excitation, which in turn triggers a change in charge in a reaction site, which is ultimately converted into chemical energy, such as the production of sugars, for the cell to store and use later as a source of energy, rather like storing energy in a battery the size of a molecule. The problem for photosynthesizing life forms is that electron excitation must be transferred extremely fast and efficiently from the antenna to the sugar-producing core before the energy associated with electron excitation is lost. Awareness of this extreme time pressure and need for efficiency triggered the very interesting idea that maybe there's something like quantum computation going on in chlorophyll to optimize energy transfer. The idea was that energy from captured photons could explore every possible path from a protein surface to the reaction center at the chlorophyll's core, and then effectively take the shortest and fastest path. To be honest, I do not know whether these ideas will end up being falsified or ultimately vindicated. However, if true, photosynthesis, which is the ultimate source of the energy that keeps us alive, would be rooted in quantum domain coherence. All living things, including ourselves, are, at the fundamental level, creatures of the quantum domain. This is simply because atoms are possible only because of the quantized states of elementary particles. If it were not for the quantized states of electrons, they would not form discrete electron shells around the nucleus, and atoms as we know them could not exist. Since we're made of atoms, we could not exist if electron states were not quantized. The key question we have considered here is whether quantum domain indeterminism matters to brain-level indeterminism. The evidence for timing differences associated with quantum domain events like Brownian motion of a neurotransmitter molecule diffusing across the synaptic cleft, or of the release of a magnesium ion from the pore of an NMDA receptor is very strong. These mechanisms alone amplify randomness from the microscopic domain to the macroscopic domain of neural spike timing randomness. And since neurons are fundamentally temporal coincidence detectors, and since the information realized in neural spiking depends on which inputs have made a postsynaptic neuron fire, information processing in the brain is surely made indeterministic by these processes of microscopic to macroscopic amplification. But in this section, we've considered the more radical idea that quantum domain coherence among non-local particles might play a role in brain function. I started out expressing my skepticism toward this idea, since so many wacko ideas have been proposed. But then I looked at the actual evidence and found evidence for a role of quantum domain coherence, particularly in systems that have evolved to detect light. I think the evidence for a role of non-local coherence in the avian retina, and in the case of light capture by plants, is increasingly strong and convincing. I do not know where else quantum domain phenomena play a consequential role in biological systems, but as I mentioned before, if quantum domain indeterminism is amplified up to a level of macroscopic biological relevance in even a single tiny corner of the universe, whether in our brains or in the brains of birds, then the entire known universe is made indeterministic. I find this thought to be a very exciting and liberating one, and I hope you do too.